dear learners. I hope you are fine. As usual, we are continuing with our topic, which is transport. But major today we are going to be looking at transpiration. So transpiration is going to be our major theme today. But still, we are under this topic. This topic is a little bit wide. Transport is a little bit wide, but we shall try to compress it. As usual, my name is Bbumba Pratrick, and feel free to ask any question that is biological, that is under biology, for the topic that we'll be learning. Now, previously, we had looked at some numbers, graph numbers. So some of you are still here. And we had, this was the graph number which we looked at previously. And I think we were able to discuss it, if not mistaken. Okay. And it had like define root pressure and then transpiration pool, explain how wind, air, temperature, light intensity, and humidity influence the rate of transpiration. Okay. So transpiration, ever since you began O level, you've been learning about transpiration. So most of these things are actually with you, you already know. But for today, we are going to see how we can actually measure the rate of transpiration using the potometer. And I hope you still recall, or you still have that knowledge on the background of your background about the potometer. But we are going to learn it today. Still, there is that question which you, if in case you want to learn more, you can interest yourself in by looking at such a question. It is good for practice of description of graph numbers, as well as explaining and interpreting graph numbers. Because if you cannot do that, that means for paper 2, you are wasting time. You cannot pass paper 2 if you do not, if you cannot study, interpret, describe, discuss, or explain a graph. Those are the crucial things to do with paper 2 biology, that is at A level. Okay. Now I will not go through that number, but good enough, you have seen it. If you are wise and maybe you need it and you don't have access to it, you can take a screenshot of that graph. Probably you will try it in your free time in case you want. For some of these graph numbers, you can always, and actually I uploaded these notes in Google Classroom. They are there, and even this number is there. For some of you are not on Google Classroom, you can always take a screenshot. And then, of course, as usual, this program is sponsored by Edify, Schools Owners Association, and then the ICT Teachers Association of Uganda. So today we are going to basically look at the internal factors. So by the end of the lesson, you should be knowing and to explain and identify the internal factors affecting the rate of transpiration. Okay. We are talking about internal factors, not external. External, you will see that most of them we're looking at environmental factors like temperature, those ones, but internal, they have to do with the plant itself. So these are anatomical, okay, and morphological. Morphology just means like form, how it is, and then anatomical, the structure and the form, or those features of the plant which influence the rate of transpiration. Okay, and there are many. Maybe for today, not to first give you a clue. Can you first tell me, or can you first help me identify any features that you think the plant plants possess that may affect the rate of transpiration? Okay, yeah, I think let me first have some people. Let's brainstorm this. I'm seeing we are having Deborah Chisache, Emmanuel, Gift, Hilary, Kadinda, all of you. Anyone, you can raise your hand and then you tell us. Yes, Letitia. Oh, let me unmute you. Yes, Letitia.
Yes, teacher, we have the cuticle of the leaf.
The cuticle of the
Leaf.
Okay, how does that affect the rate of transpiration?
When the cuticle is waxy, it covers that covering on it reduces the water loss, the surface area for water loss.
Okay, thank you very much, Letitia. Okay, yeah, that is actually true. It minimizes. Okay, thank you, Letitia. Then gift. Which other gift?
The leaf surface area and surface area to volume ratio.
What do you mean by surface area?
Okay, like let me say, like if the leaf is large or small, it can also it can affect transpiration in a way that the more bigger the leaf, I think the more bigger the leaf, the more water loss, yeah, that affects it. Water loss is easily like water loss, how can I explain it? Like if the leaf is large, then transpiration can easily take place.
Why?
Why?
Why is that? When the leaf is large, then transpiration may occur more often.
I think so.
Okay, thank you. Let me. I'm seeing Lucky Champ. Yes, Lucky Champ.
Okay. The number of the stomata in the leaf. The more the number of stomata, the higher the rate of transpiration.
Okay.
And also, yeah, you're welcome.
Okay, that's very good. Then Emmanuel.
Opening and closing of the stomata. For example, it opens during night and it closes by daytime.
Okay, that is a stomatal rhythm. Okay.
Yes.
Yes, Joseph. Any other?
Hello, good morning, everyone.
Yes, good morning, Joseph.
Yes, I came late, but I don't know whether they talked about the cuticle.
The cuticle, yeah, someone had talked of a wax cuticle.
Okay.
Yeah. So and so on and so forth. Are you guys exhausted? Are those the only features that you can identify? Yes, Joshua.
Yes, teacher.
Yes. Which other?
I want to clarify more on the point of the stomatas.
Please go on.
If, on a leaf of a plant, there are more number of stomatas at the upper part than the lower part of the leaf, and this affects transpiration such that it doesn't lose a lot of water.
So you were saying that the upper part of the leaf has more stomata than the lower side?
Yes.
Okay, Joshua, I want you to take note of that. We shall see whether it is the other way around or not. And then, yes, Luck. It is what?
Okay,
it is the other way around.
Okay, yes, Lucky Champ.
Okay, Master, I wanted to talk about this point of the thickness of the leaf.
Yes, go on.
The thicker the leaf, the lower the rate of transpiration. Reason being, the distance at which the diffusion takes place. The thinner the leaf, reason being the rate at which the water the length at which the water diffuses is shorter.
Okay, thank you. Well, of course, you're all giving good answers, although some need to be harmonized. Let me first read some here in the chat box. I'm seeing Sandra is saying that number of stomata, but she didn't say much about it. Then Raymond says number of stomata on leaves. Then Lucky Champ says the leaf thickness. Here I say sunken stomata. Yeah, I like that answer. Sunken stomata. We shall see what we mean by sunken stomata. Let me also last person here, Deborah Chisache.
Teacher, good morning.
Good morning, Chisache.
I wanted to talk about the sunken stomata.
Yes, please go
on. For the sunken stomata, they are hidden, so they are not directly exposed to the surface, and so they trap some water vapor, and thus reducing the concentration gradient of water between the air and the leaf. As such, they are reducing the rate of movement of water, reducing the rate of transpiration.
Okay, thank you, Deborah. Yeah, so okay, let me end with Joshua Kandinda, and then we begin. Yes, Joshua?
Yes, teacher. I want also talk about the mosaic arrangement of the leaves on the plant.
Where please go on.
Those leaves that we call the shade leaves are under other leaves on the plant. They end up transpiring at a very low rate.
Okay.
And those that we call the sun leaves that are easily exposed to the sunlight.
Okay, yes, thank you, Joshua. But the mosaic leaf arrangement may not be specifically for, you can't group it specifically that between sun or shaded plants or those that are non-shaded. Both of them could have a mosaic leaf arrangement, actually, as we shall see. It doesn't have to be whether a sun leaf or a shaded leaf. Okay, and then Omenya. Yes, Omenya. Raymond.
Yes, good morning, sir.
Morning, Raymond.
Yeah, I wanted to talk about, like, some plants have airs on the surface of their leaves that attracts the water vapors on the surface of the leaf.
Raymond, does it attract or it traps?
It traps. It traps the water molecules on their surface that reduces the rate of transpiration in such a way that the diffusion gradient will be lowered.
Okay, thank you, Omenya. Okay, I think to keep on going, you guys know these things. I think, and for today we shall not even waste time on these internal factors, because the answers you are giving are actually very, very good, although some just need a few harmonization here and there. So one, the leaf area, which you guys have already talked about, I think it was Rita. So the greater the total leaf area of a plant, the greater the rate of transpiration. And why? And then you find that flat and broad leaves have a greater rate of water evaporation from their surfaces than smaller ones. And that's why, as a way of reducing the rate of transpiration, some plants tend to grow smaller leaves compared to others which are found in places that are waterlogged. When you find plants that are found in waterlogged areas, they tend to have broader leaves to increase the rate of transpiration. Okay, and then also the cuticle, which you guys have already talked about. The thicker this cuticle, the lower the rate of cuticular transpiration, because we have about 3 types of transpiration: cuticular, then stomato. Because not all water is lost through the stomata, as we commonly know. Some can be lost over the cuticle. Then the cuticle is a waxy covering on the leaf surface, which reduces the water loss. And then another one is the density. Now this one you hadn't talked of it from all the people that I had chosen. The density, that is, you're looking at the number of stomata for a given leaf. So the higher the transpiration rate. So the lower leaf surface, that is the abaxial side, possesses more number of stomata than the upper side, which is the adaxial side. And I think this is what Joshua was talking of, but he was reversing. He was saying that the upper has more and then the lower has less. So it depends on the plant, but most of the plants will have the lower side having more stomata, such that stomata are not directly in contact or in direct interface with the sunshine, and that minimizes the rate of water loss. So the evaporation of water takes place more at the lower than on the upper. And there's an experiment which you guys used to do in O-level, whereby you would put cobalt 2 chloride paper on the underside as well as on the upper side. And you find that the pink cobalt 2 chloride paper, which is put on the underside, would turn to blue quickly, just because it tests for the presence of water. Joseph says that even lenticular, yes, that is true. As I said, that there are about 3 types of transpiration, which is cuticular, stomato, and then lenticular through the lenticels. Thank you, Joseph, for that. Then Emma says that, sir, I thought it depends on where the plant has grown, Iggy water plant. Yes, that's what I say. It depends on the type of plant, but most of the plants will have more on the underside. You get it? But it depends, as Emma, you have said, it depends on the type of plant, but most of them will have more on the underside. And then you see here, now this is, we're having picture A here, then we are having B here, and then C, and then D. Okay, and you can see that you could have differences in the number of stomata. Look at this picture A. Okay, here, this is a different plant. Well, this looks like the plantago plant. Then you're having this other plant, then this other again plant, and then this other. This looks like grass. You can see here, and you can see the type of stomata that they have. Actually, for the last 3 years in biology practical, questions that have been coming, I think the last 2 years they have brought the commelina, the real commelina, the one with the red protoplast, whereby they tell you to peel the underside so that you can be able to look at these stomata in the microscope. But of course, last year's UNEB, instead they did not use the stomata, they used these other cells, epidermal cells, rather than the stomata. Because when you are looking at this, you may observe these other epidermal cells, or you may observe the stomata. So a question can come either way, whether you are looking at the epidermal cells or the gut cells, depending. Then there's the one which came in 2003 UNEB. It was looking at stomata aperture, whereby they put solutions. You had 3 solutions of different concentrations, and you had to put these epidermal tissues inside these different solutions. One was distilled water, another one was a solution of about 3 molar sucrose solution, and then another one was having about 1 molar sucrose solution. This was just distilled water, which is 0.0 molar sucrose solution. You get it? Solution. So we find that the one placed in distilled water is going to open its stomata widely, and then the one placed in about supposed to be 0.3 or 0.25 molar sucrose solution. Now this 0.3 or 0.25 molar sucrose solution is actually the same concentration as the sap of the guard cells. So you can imagine what could happen to the stomato aperture here. And then the one placed in 1 molar sucrose solution loses all the water and the guard cells close. And they were told to draw these different stomata from the different solutions. So you must have that knowledge there as far as practical is concerned. Also distribution of stomata, which we have just already explained. And then normal, of course, this is how they appear. They are kidney shaped, or you can say that they are bean shaped. And then Joshua says, is that osmosis? Yes, Joshua, what is osmosis? What do you think osmosis is? What do you think osmosis is? Okay, so yes, Joshua.
Osmosis, it is the movement of water from a point of high concentration to a region of low concentration through a impermeable membrane.
Very good. Okay, so once you see that there is movement of water from one side to another, then just know that that is osmosis. If it is not water, yes.
I thought that on that question that came in 2003 UNEP, that those solutions that were being pressed on the upper surface, on the parasite surfaces of the leaves, that because of osmosis, that's why the cells would open due to different concentrations of sucrose solutions.
Yes, yes.
Yeah.
And what was your suggestion?
So I wanted you to clarify me how transpiration would come probably with osmosis at the same point in that question that came.
Hey, okay. I think there I just went off a little bit. I was saying that the reason as why I emphasize this, it's not because of transpiration. This is osmosis, but I brought in this question of 2003 UNEB to make sure that you guys are aware that you are supposed to know how to draw these guard cells. Okay.
Okay. Yes.
You are supposed to know how you to draw these guard cells. That's why I brought it. But it's not that they are going to change in these different concentrations because of transpiration. No, don't get me badly. Okay. This is pure osmosis. This is pure osmosis. Okay.
Okay.
So and then in other plants like the oak and apples, they limit stomata entirely to the abaxio, that is the lower surface, to reduce overall water loss. Actually, when you look at most plants, there are some plants without actually no stomata on the upper side. There are some of them. And when we examined actually that Rio Comelina, I don't know if they've ever seen it in the lab. You may find like 1 or 2 stomata on the upper surface. You get it? Then there are also some other factors which affect the temperature of the leaf, which include sprays and dust, which affect permeability of the cuticle and temperature of the leaf. Especially plants which are on the roadside of dusty roads, or people who spray crops with certain insecticides and all those pesticides. Then also, you can see that we're having a small graph here, which looks at the rate of stomatotranspiration. They are increasing. And then here you're having diameter of the stomato aperture. Diameter of the stomato aperture. So you find that actually, if you are keen enough, they can act like this, and they ask you to kind of showing how the rate of transpiration is going to increase in the wind area or environment, and then this one is showing how it is going to increase in still air. You can see that. So we can now look at measuring the rate of transpiration. Now, when we are measuring the rate of transpiration, it is actually, we measure the rate of water uptake using a potometer. But because the water taken up, we tend to say that it is somehow correlating with the water loss, because it is after water is lost than that water is being taken up by the plant. But it should be noted, and I repeat, it should be noted that it's not true that the water that is lost over the leaf is the same as the water that is taken up. Reason being, some water may be used in some other processes within the plant, such as photosynthesis. You see that? So that's why it's not entirely, but of course it gives us a picture of trying to calculate the rate of transpiration by looking at the water loss. Otherwise, you cannot collect this water that is lost over the surface and then you measure that. That wouldn't be feasible. Now, there's a video which I'm going to show you right now about the rate of transpiration. Although this one did not use this normal potometer, which we know. Now, the normal potometer normally will have where you place the plant, you fix it here. This is the leafy plant, okay? And then it will have a reservoir. This reservoir will help you to push back this dot, this air bubble back to the starting point here. And then also, this is a water where you can place. Now, this one is a little bit a different model, which is not the one of the potometer, but it's going to help us understand very many things. The video which I'm going to watch, help you watch. I was supposed to actually embed that video in this PowerPoint, but the problem came with it becomes slow. I don't know why it was becoming slow, and that's why I decided to play it direct from here on the screen. Okay, now let me play this video. Okay, I hope you guys can be able to hear the voice from the video.
Yeah, we can hear.
Okay, thank you. Let me play this video. Okay.
This video will take you through how to measure the rate of transpiration of a plant using a potometer. Before we start, this is essentially what you're aiming for, but the most important piece
now
of
this is a different model.
is to
You can
make
see
sure
that
it's
this is the leafy
airtight
plant
with no air bubbles in it, and that's
then
also
they
the
have
most difficult
connected
part
this
of this.
capillary tube to this kind of a scale, okay, which is actually when water moves on this scale, as water is moving, you are able to read, it has a reading here, the amount of water that has been taken up. Because as water is lost over the leaf surface, it keeps on reducing and falling up to, and you can even read different values. But for the case of the disadvantage with this kind of setup is that when you want to take it up, you have to redo it again, which is very much tiring, okay? But for the potometer, it has a reservoir, okay? Joshua says, is that the reservoir? No, this is not the reservoir. This is actually what is going to read. It has the scale. If I can annotate a little bit here. These are values. Those are scales there. It has those values on it. So let's watch the video. Try to be keen enough because, of course, he's speaking like the English. I hope you try to get his accent.
Setup. The first thing you want to do is take a glass pipette and submerge it in a tray full of water. If you tip it slightly, you'll notice that all of the air bubbles come out of the pipette. If they do not, you can gently use a plastic pipette to suck the remaining air bubbles out of that pipette. You want to ensure that no air bubbles remain. Once you have done that, you want to repeat the process with the plastic tubing, again tipping it to remove the air bubble plastic pipette to suck the remaining air bubbles out of that pipette. You want to ensure that no air bubbles remain. Once you have done that, you want to repeat the process with the plastic tubing, again tipping it to remove the air bubbles and, if necessary, using a plastic pipette to remove the remaining air bubbles. Now, whilst this process is tricky, you want to make sure that you really spend the time making sure that you get the air bubbles out of the glass pipette and the plastic tubing. If any air bubbles remain in either of those, then the potometer will not work properly. Now, in the spirit of keeping air bubbles out of your potometer, the next piece has to be done very quickly. You need to cut the stem underwater. Now, ideally, the whole plant would be underwater, but clearly if you have a pot plant with soil, that's difficult. So just do it as quickly as possible to avoid any air entering into the xylem of the plant. Once you've done this, you can take the stem that's attached to the leaf and insert it into the plastic tubing. Take the plastic tubing and attach it to the glass pipette. Of course, all of this still needs to be plant. Once you've done this, you can take the stem that's attached to the leaf and insert it into the plastic tubing. Take the plastic tubing and attach it to the glass pipette. Of course, all of this still needs to be done underwater to continue to avoid air bubbles from entering your potometer. It is often recommended also to use Vaseline at the points of connection in order to ensure an airtight seal. Again, continue to do all of this underwater until you can be as sure as possible that your seals are airtight. The aim is not to have any air bubbles in the apparatus, and this will allow for the greatest chance of having a successful working potometer. If you have any parafilm available, this can also be extremely useful in helping to secure those seals. Once everything is as airtight as you possibly can make it, remove the potometer and gently attach it to a clamp and stand as shown here. At this point, you might notice some air bubbles in your potometer. If that's the case, it may be that you might need to start from the beginning and remove the air bubbles again. To measure the rate of transpiration, first mark where the water level is on the glass pipette. In other words, where the air has gone into at the end of the glass pipette. During transpiration, water evaporates from the leaf. As a result, water is taken up by the plant and the air moves into the glass pipette from the open end. To measure the rate of transpiration, you'll need to measure the distance that air bubble moves into the glass pipette in a given time. The greater the distance moved by this air bubble in a given time, the higher the transpiration rate. It is possible to use this potometer to look at the effects of different independent variables. For example, a fan could be used to simulate wind. The different speeds of the fan to simulate different wind speeds. You should compare this against a control where there is no air blowing or still air, represented by turning the fan off. You should also be mindful of controlling other variables. I have seen students before put a bag over a leaf in order to simulate still air, but be aware that this would alter the humidity surrounding the leaf. So, as a review, the potometer works because water evaporates from the leaves during transpiration, and therefore the air bubble will move into or along the pipette as the water is taken up into the plant to replace that lost through evaporation. In order to measure the rate of transpiration, measure the distance or the volume into the pipette moved by the air bubble in a given period of time. Make sure that you repeat your measurements and control for other variables. For example, when you repeat it, keep it at the same temperature. Then once you've measured the distance or the volume in a given period of time, you can calculate the same temperature. Then once you've measured the distance or the volume in a given period of time, you can calculate rate by doing distance or volume divided by time. The units, therefore, might be, for example, milliliters per minute or centimeters per minute. A word of caution. Measuring the rate of transpiration using a potometer is an indirect measurement, and this is because it measures water uptake as opposed to the water evaporating from the leaves. Be aware that water that is taken up by the plant can also be used for other processes, for example, photosynthesis. So the actual transpiration rate may be lower than that indicated by the pedometer.
Okay, I hope you've been able to follow that English very well. Okay, let me first get my pen. Yeah, so you've seen that has been a slightly different model of a potometer, but of course, for us, this is the model which is very simple. We learn. Okay, so the leafy shoot is placed in the upper xylem inside, space within the xylem vessel. Because if you cut it up, not underwater, some air bubbles may enter in the xylem and affect the results. Then also the rate at which the bubble, that is this bubble, moves can be recorded by measuring the distance on this graduated capillary tube. Graduated means that it is having measurements on it. The measurements are clearly stated, and you can actually read it. You could find that this is 0, 1, 2, like that, 3, 4. So you can even actually measure the distance moved by the air bubble. Okay. And then 3, water from this reservoir can be left into the apparatus to push the bubble back to the starting position by opening this tap here. Because I'm assuming this bubble had moved up to here. When you open this side, and you can restart the experiment. So that means measuring the rate of transpiration has the following procedures. So first of all, you must be able to label this diagram. And I think you can understand it very well. I used to have a video, yes, that I looked for it and I lost it. I don't know where I put it, but it was having this very experiment being done. Now you cut a leafy shoot of a plant. Rodney says that, Master, I am requesting you to please help me with the class code for the Google Classroom app. That one, the admin will send you the code for the Google Classroom app. Then here, cut a leafy shoot of a plant. If possible, hold the plant where the cut is to be done underwater. This is what the lady has been doing. This is to prevent air to enter the xylem. Then the rate of water uptake is measured by introducing an air bubble at the end of the capillary tube, timing how long it takes to move between the successive divisions on the graduated cylinder. Here, you can actually time very well. What we are actually revising is what the lady has been doing. So when one set of readings has been obtained, the air bubble is returned. So after returning one set, and normally remember the rate of transpiration is measured in distance per time. So you get the distance covered by the bubble divided by the time. You must have a stop clock to measure time either in seconds or in minutes, depending. Okay. So, and then also the results obtained expressed as distance moved by the air bubble per unit time, which time can either be measured in minutes or in seconds. And the apparatus can be improved by being calibrated, because you may not, if you don't calibrate, that is, you clearly state the demarcations or markings for the distance. So by estimating the volume of water present in a given length of the capillary tube and the leaf area calculated with square papers here, that is to increase it in case. Now in this case, uptake of water can be expressed as volume per unit time. But in most cases, what we do here is we express it in terms of distance per unit time. And then to measure changes in the rate of transpiration with various external conditions, the experiment can be repeated under different conditions. Those are the conditions that the lady did when she brought the fan to represent moving air or wind. Or in light, you can actually bring a bulb close to this this leafy shoot, all in the dark, or you can change temperature or humidity by introducing the other polythene bag, or in still air. So however, the results obtained is not being accurate measure of the rate of transpiration because a little water is taken up and is used during the process of photosynthesis. This is what the lady was also explaining in the video, okay? So if you have understood, we're having a small question here to kind of make us see, to assess us and see whether we have understood. And the question is very simple. It says that the experiment was carried out under the following environmental conditions, numbered 1 to 5. So still air leaves untreated. And then moving air using a fan leaves untreated. Then still air leaves Vaseline on upper sides only, like using Vaseline on the leaves. Then also still air leaves Vaseline on the lower side only. Then still air leaves Vaseline on upper and lower side. Temperature and light intensity were held constant throughout the experiment. And the first question says that state precisely what physiological process the apparatus is designed to estimate. The apparatus which we are looking for, which we are looking at, is this. This is the apparatus here. I think you have looked at the apparatus. Now the question says, state precisely what physiological process the apparatus is designed to measure. What would be the answer? Yes, Deborah?
So it would be transpiration.
Very good. That would be transpiration. So the answer here is transpiration. Okay. Then part B says the results are identified by the key letters V to Z, but they are not necessarily in the order in which the experiment was conducted. Copy and complete the table below to indicate in each case which of the conditions 1 to 5 produced the results V to Z. So we are having results V, W, X, Y, and Z. And here we are seeing distance measured in millimeters moved by bubble after 120 seconds. The conditions are the ones which we can actually identify. This is distance 3, 10, 150, 45, and then 60. And then we need to know the conditions now. I don't know whether you understand this question. Let me not first interpret it for you, because question interpretation is also part of A level. So what do you think we are supposed to put here? Okay, because some people it may seem obvious, but people may fail to understand this. So let me first have someone raise their hand and you tell us how are we going to answer this question? Sandra. Okay, let me unmute Sandra. Yes, Sandra.
So I think we fill in the condition.
Sandra, you think we fill in conditions? How?
Conditions that have been given in the question from 1 to 5.
Like by writing here, still air leaves untreated here. Sandra?
Yes, teacher, I said, I think we fill in the conditions that have been given in the question earlier on from 1 to 5.
So you mean I come and I write all of the, for example, condition 1 says still air leaves untreated. So it's what I write here?
No, you study them, then you look at like the distance in millimeters moved by bubble after 120 seconds. So you look at which condition could suit that very distance covered or moved by the bubble after 120 seconds.
Okay. Okay, thank you, Sandra. Okay, I think, yeah, but as you can see, the question is very simple. Says, copy and complete the table below to indicate in each case which of the conditions 1 to 5 produce the results. So if you think that, for example, this distance moved in 3 millimeters is supported by condition of still air, leaves untreated, then you come and put here 1. You see that? When you see, maybe the answer here is 4, you come and put here 4. So now I want you to give me the proper answers. Let me first erase off this. So what do you think is going to be for the first? Wait, let me first get my pen. Let me get my pen. Okay. Who was first? Let me choose someone. Now, for some of you who have not named yourselves, like Samsung, please rename your gadget, because I can't call now Samsung to answer. Samsung is not Uganda. Let me choose here. Emma Emmanuel. Emma wrote Emmanuel. Yes.
So for V, I think it's 5. Still air leaves Vaseline on upper and the lower sides. I think it's
Here.
5.
Okay,
5.
so according to Emmanuel, this is 5. Okay. How about I'm seeing Lucky Champ. Lucky Champ, what do you say?
Okay, Master, I would like to give all the answers because I've gotten all of them.
You only give us 1. Do you agree with the first one?
Yeah, I agree.
That Emma has given?
Yeah, it's 5.
Oh.
The next one is...
The next one is what?
4.
Like your network is not favorable. 4, eh?
4.
Okay. Lucky has 4 here. Okay, thank you. Let me choose another person. Okay. What about the rest? Rudini Seth? Yes, Rudini.
The next is 1. The next is 2.
On X is 2, eh?
Yes.
Okay. Let me see. Then I'm seeing Mildred. Yes, Mildred. Mildred, we can't hear you. We can't hear you.
Hello?
Yes, we can hear you now.
Y is 3.
Okay, Mildred says Y is 3. Okay, now let me choose another person here. Joseph. Yes, Joseph.
Z is 1.
Okay, Joseph says that Z is 1. Okay, do you guys all agree with these results that people have suggested? Or there is someone, if you feel like you want to change anyone, let's say V, W, X, Y, and Z, you can tell us that for me, I'm not contented with this, and you raise your hand. Okay, I'm seeing, yes, Joseph, what do you want to say? You're not contented with something there?
Teacher, this That one was out on W. To my
Pardon?
side. Hello.
Yes.
That value of W. I think it should be 3. That is my idea.
So this one thinks this should be 3. And then how about, okay, let me choose Samsung here. I said that he doesn't know how to name. Yes, Samsung. You may tell us your name. What's your name, Samsung?
Yes, sir. Good morning.
Morning, Samsung. What's your name?
My name is Aparango Cinderella Nomo.
Okay, Nomo.
Yes, for me, I don't agree with the V. I think that it should be 4, condition 4.
Okay, Nomo says this should be condition 4. Okay, thank you, Nomo.
Yes.
Cinderella.
Can you help me?
Pardon?
I am asking that if you could help me and rename myself on the
Hey,
phone.
okay. Okay, I'll do it for you, but not now. We are left with a few minutes.
Okay, thank you.
Then we're having Emma wants. Okay, let me first choose Omenya. Raymond. Yes, Raymond.
Yes, sir. Like for me,
your hand is up. You want to do that?
Yes, from my side, I agree with what we did the first time. That is V to B5, W to B4, X to B2, then Y to B3, and Z to B1. The reason being, seeing W first, we choose 4 because the Vaseline was applied on the lower side of the leaf. That has more stomata than the upper side of the leaf. So, so it's like this. It means W will lose what? The condition 4 will lose more water.
Okay, okay. Thank you for that. Mute. Let me first mute this one. Is this organizing our lesson? Okay. Someone was disorganizing the lesson. Okay, let us try to analyze. Let us try to analyze these answers. Joshua says the first conditions are actually correct. Let us see. We are looking at these conditions. You're having still air, leaves untreated. Okay. Here, untreated, we mean that those leaves have not been smeared with anything. They're just in their natural conditions. Then here we are seeing moving air using a fan, leaves untreated. Now, when you're having a fan blowing any water vapor that has just escaped, it means this one probably should move the greatest distance because the leaves are not treated and there is a fan that is pushing. You see that? So when you look at this, which one is the greatest distance covered? Okay, you can see this is the greatest distance. That is going to be X. So that is true. We may, okay, let me get a blue pen here. Okay, where are you, my blue pen? Okay, so we may agree with this. Then still air leaves Vaseline on upper sides. Okay, and the rest have still air. So now we begin. So far we have eliminated this one. Okay, and then we are having still air leaves untreated. Then we are having still air leaves Vaseline on upper side. So this one should be lower than than 1. 3 should have moved a shorter distance than 1. Then still air leaves Vaseline on lower side. Also this one. Then still air leaves Vaseline on upper and lower side. So that means one should go on the next distance. Okay, that is correct. So we can also remove this. Then still air Vaseline on upper side, and then we're having still air Vaseline on lower side. Since the lower side tends to have more stomata than the upper, so we are going to take this as the next bigger distance. Still air leaves Vaseline on upper side only. On upper side only. That is going to be 3. Okay. Then, okay, we are done with that. Then air leaves Vaseline on lower side. And then still air leaves Vaseline on upper and lower. So this one, since it is only the lower side, we may consider it as the next distance moved. Next distance moved is going to be 10. Okay. And then still air leaves Vaseline on upper and lower. So this one should move the lowest distance. Okay. Thank you, guys, for that. As you can see, it is time up because our lesson is always 1 hour, but we are having these other questions. So I would like to say goodbye and enjoy yourselves because time is up. Bye.
Thank you, teacher.
Bye.
Thank you,
Bye.
teacher.
Thank you so
Bye.
much.
You're welcome.
Bye.
Thank you.
Bye. Bye-bye.
Bye-bye.
Thank you.
Okay, bye-bye.